Today I would like to explain some fundamentals of our base editing systems. I don't talk so much about the therapeutics, but I would like to show you the fundamentals of these editing systems. So probably, as you know, the CRISPR-Cas9 cleave the genomes, but our systems, base editing system, is non-cutting genome editing systems. So that means this system is like target AID, target G. This is a base excitation reaction linked to DNA binding motifs. So in that sense, this is actually doesn't cleave the DNA. So in that sense, this is very mild systems. So the system, total system, looks like this: cytidine, deaminase, and the glycosylases, and more. These kind of base editing enzymes linked to the dead or nickase Cas9 systems. So this is a basic way of the basic concept of these systems. So actually, the idea comes from like this, So if you think about this, Vetebtate adaptive immunosystems, usually this system, AID, activated induced cytidine deaminase systems, that create a mutation to diversify the antibody specificities. So in a sense, probably you understand this might be very specific just to create diversity of the antibodies. So actually, if you look at about these systems, this AID just function in a single-stranded area. So and if you look at this bacterial adaptive immunosystems, it's of course CRISPR-Cas9 systems. Here we have a guide RNA, and here we have Cas9 systems. But if you compare these two systems, it looks kind of similar. So that is the origin of our concept. So then we conjugated these two systems, dead or nickase Cas9 linked to the AID. So then you can imagine that this AID is just active to this single-stranded area caused by these guide RNA systems. So in a sense, basically, you can expect that this also must be very specific, editing systems. So we chose this AID from the C-Lamprey systems because that is very primitive Vetebtate, and we expected that this AID is very high, has high activities. So that is a very basic concept, and this is how the our editing system works. So this AID, DNA deaminase, caused base change. That means the cytidine. Here we have amino groups, but by removing these amino groups by the AID systems, this cytidine changed to the uracil. So then uracil has a base pairing property of thymines, T, like T. So as a result, this kind of editing reaction happens. So deaminase changed the C to U, and then this U is has the same base pairing property as thymine. So opposite side of this U must be A. And the U is unusual, so finally U changed to the T. So like this, by using these deaminase systems, we can create the C to T change in a very specific targeted area. So that's the reason why these systems are called as pinpoint genome editing systems. But of course, life tried to repair these mutations by so-called base excitation repair systems. So first of all, so uracil glycosylases remove this U, and then AP side cleavage happened, then create a nick, and this repaired to the original base. So like this, during this mutation, the life tried to repair. So as a result, not just the C2T, but the C2TAG could happen. But actually, if we use this base editing systems, most of the mutation is just the C2T. So in the sense, this is very specific mutation systems. But depending on the life system, this repairing mechanism characteristics are slightly different. As a result, depending on the life system, a variety of different phenomena we can observe. Today, I would like to explain about these kind of very fundamental things. So first of all, we try to construct most suitable base editing systems. So in a sense, we compare the variety of the configurations. So this, we just look at this 1, 2, 6, and this is standardized mutation abilities, and this is on site, on target, and this is non-specific part. So if you look at 1, 2, this is just a deaminase, AID, or DCAS9. Of course, nothing happened. But if you look at this one, surprisingly, this is DCAS9, and deaminase, AID, is just expressed separately. But you can already see the high efficiency of the mutation. So, and then if you look at the 4 and 5, 6, 5 case, this link to the deaminase linked to the by using the short linkers. So in this case, the efficiency of mutation is lower. So in that sense, we need a kind of enough length of the linker systems. But if you compare these free deaminase systems and these conjugated systems, the efficiency is kind of similar, so surprisingly. So like this, AID need not necessarily to be conjugated to the DCAS9 systems. But our cases, we utilize this conjugated system 6 with enough length of the linker systems, because this is more easily we can utilize as a one protein fusion proteins. So then we compare the variety of the this Cas9 part. This is just Cas9, and this is Nikkei's Cas9, and this dead Cas9s. So Nikkei's Cas9 case, we have 2 options. In this case, we have activities to the same site, site of the deaminase, deamination strand. But in this case, this D10 to A Nikkei's cases, it's active to the opposite side of the deamination site. So, and this is a DCAS9. So, first of all, we compared which construction is most, more efficient. So this is the results. So just look at this one. This is this Nikkei's case, D10A Nikkei's, which create a nick to the opposite strand of the deaminations. This is the same strand. So if you compare these constructions, you can clearly see this efficiency is much higher than the others. So that means this strand, this construct, so Nikkei's, which create a nick to the opposite side of the deamination, is much higher activity than the others. So if you think about the reason, probably reason is something. This is just a hypothesis. So if you create a mutation here, so if we have a nick here, so life try to repair this by removing this and using this deaminated side strand as a template. So in a sense, we can have a much higher mutation efficiency by using this Nikkei's Cas9, and which create a nick to the opposite side of the deaminations. So like this, we can optimize the efficiency of the systems. So under this slide, okay, so I forgot to tell, these experiments are done by utilizing the Saccharomyces cerevisiae as initial studies. So this slide is a very important slide. So this is a guide RNA sequences, and this is Cas9 systems, and this is dead Cas9 deaminase. This is a Nikkei's Cas9 deaminases. So in the case of the Cas9 case, this is a mutation, and this is a deletion, and this is insertions. So like this, in case of Cas9 case, variety of things happen because double-stranded break happens. So after that, life tried to repair in a variety of types. But if you look at this deaminase case, here we have a C here. It's, sorry, very small. But, just a single nucleotide change, just that happened. Nothing else happened. So in a sense, we can say these deaminase base editing systems are very clean, editing systems compared with the Cas9 systems. So like this, we can, utilize this system for the more specific point mutations. So in the case of the using the Saccharomyces, we try to clarify the, mutation spectra. If there are many C in this target area located, so which C is most, preferred for the deaminase systems? So like this is a different sequences. Like this, around -18, around that area, the frequency of mutation is highest. Of course, if you have a variety of C nearby, of course, there is some spectra. But around this 19, we have the highest mutations. So in the case of the yeast cell, this is a very specific case. So this, if you look at the change of the bases, not just the C2T shown by the yellow, but also C2G and C2A, mostly C2G and C2A happened. This C2G mutation probably caused by the BER reactions. So like this yeast case, this is very special case, in addition to the C2T, C2G mutation can happen. So we can summarize like this. So the mutation spectra is like this. Around the -18, this area, we have a very narrow window for the mutations, and mostly C2T mutations. But in the case of the Saccharomyces cerevisiae, this is a special case because of special BER mechanisms. In addition to the C2T, C2G also can happen. So like this, there are very specific mutation spectra and mutation tendencies. So at the same period, we published these papers. David Liu teams in Harvard University also published a very similar result. So, and they are using the UpVec systems, and we are using this Silampli systems, and the mutation spectra are slightly different. That means that depending on the AID systems, deaminase system mutation spectra are varied. So you can adjust the mutation spectra by choosing the appropriate AID systems. But don't worry, we are not fighting. That's pretty important, So David Liu teams and our team try to collaborate. So in a sense, that's helped a lot for the many pupils. So in a sense, depending on the systems, this is slightly, this spectra have changed. So, but anyway, I could say this systems works as a very pinpoint or very specific base editing systems. So, but we then move to the animal cell system, CHO cell systems, and then very unexpected things happened. So this is a Cas9 case. Of course, indel are majorities of the editing. But if we utilize Nikkei's Cas9 AID systems, surprisingly, there are a lot of indel in addition to the C2T mutations. This is very unexpected results for us. So then if you look at these repair systems, so probably this kind of things happen. So after this nick insertion of the nick, of course, this BR reaction happened. So after AP cleavage, here also we have a nick here. So then the double-strand break can happen here. So after that, by the non-homogeneous end joinings, probably indel can happen. So like this, in the case of animal cell, probably this root is much stronger than the yeast cell. So in a sense, as a result, this kind of the majority of the edited results are indel formations. So then, of course, we try to stop these BR reactions by introducing the UGI gracyl glycolase inhibitor systems. So as we expected, if we utilize the Cas9 deaminase and UGI, so we significantly reduce the indel formations. And as we expected, C2T mutation in the targeted areas, a majority of the edited results. So, but of course, if you use the dead Cas9 systems, this mutation efficiency is much lower than the Cas9 systems. So, like this, in the case of animal cell, probably in a human also, this Cas9 AID and UGI system is most effective systems. So, like this, because the repair system, characteristics of repair system are different depending on the live systems. So optimum, these kind of optimum editing systems are different. So, so in the sense, UGI increase the C2T conversion and suppress indel caused by the this Cas9 AID systems. So then if you try to this one for the in therapeutics, size of the editing system are pretty important. So this Cas9 system always already has a 4 point something kilobase size. And then this AID system also has a 800 base pair. So in addition to make a fusion proteins, so you can utilize endogenous AID. So this results show that. So in addition to this vector systems, adenovirus vector Cas9 systems, we added the interferon to induce the AID. So then this table compared. This is a Cas9 AID systems. And this is, just adenovirus systems and interferon case. So then the result of the editing is almost similar. So like this, in addition to the fusion, systems, we can utilize the endogenous AID system if you want. So like this, you may decrease the size of the total editing systems. So this is a very different systems. Probably you are not interested in, but this is an application to the plant cell genome editing case. This is a case of the gain of function type mutation case. So in the case of the plant cell, so this ALS, this protein, if you create a A96-V mutation, this plant cell have a herbicide tolerance. So that's very easy to analyze the successful mutations. So we utilize the genome editing system to create this A96-V. So just this C2T mutations. So we actually did this, and the result is like this. So dead Cas9 case, it doesn't work so well. But if you utilize Cas9 AID systems, we successfully create the mutation gain of function type mutations. So like this, I think these pinpoint mutation systems are very effective for to create just gain of function type mutation by just the base exchange. So I would like to add one more case. This is a genome editing of the bacteria, like E. coli case. So depending on the live system, this dead Cas9 is okay, but this Cas9, this is the most effective Cas9 case. This is this CFU by microgram of the systems. So then low value mean that high toxicities. So like this, similar to the Cas9 systems, so this Cas9 AID system are very toxic. So, but if you utilize the dead Cas9 AID system, this is not toxic at all. So then create a very high efficiency of the mutations. So this is because of the very poor of the or lack of the homologous end joining repair activities of the E. coli systems. So like this, what I would like to say is this repair mechanism or this kind of the repair mechanism are quite different depending on the live systems. So in a sense, suitable editing systems are different depending on your target live systems. So by using the E. coli systems, we further clarified the characteristics of our editing systems. So we utilized the different lengths of the guide RNA. This is 20 and this is 21 guide RNA lengths. And this blue showed the dead Cas9C deaminases. And this, yes. And this red one showed the dead Cas9 and the AID and UGI case, uracil glycosylase inhibitor case. So you can clearly see the differences. Two things we can observe. Depending on the guide RNA length, mutation spectra are different. And if you utilize the UGI, almost the saturation mutation happens. So that means a very much higher efficiency of the mutation compared with just DCAS9 and AID. So UGI increase a lot about mutation efficiency. So we further clarified this editing system by utilizing the E. coli. And this is the effect of the length of the guide RNA. So this is from the 19th to the 20 something. Sorry, I cannot see the exact numbers. But depending on the size of the guide RNA, so this different color shows the difference of the guide RNA. So you can clearly see if you utilize the longer guide RNA, mutation spectra are wider. Probably you can imagine that if you utilize a longer guide RNA, R loop created by the guide RNA is bigger. So in a sense, we can edit a wider region of the your target area. This is all like this. This is different target, but all have many C in the targeted area. So like this. Bigger, longer guide RNA create a wider range of the mutations. So like this, we can control this kind of the mutation spectra by designing your systems. So another very unique point of this deaminase case is multiplexities. So you can introduce many point mutations at the same times. So in this case, this is an E. coli case. We try to test how many multiplex editing are possible. So in this case, we utilize the transposase gene. In this case, we use the 4 different sequences, and totally this transposase have a 41 copy in a genome of the E. coli. So by utilizing these 4 guide RNA, we can actually introduce the 41 editing at just one time of this kind of editing systems. So AID, and here we have 4 guide RNA. So like this, because it doesn't cleave the genome, so you can create a mutation in a variety of parts at the same times. So this clearly shows this is very, very mild and specific editing systems. So in a sense, target AID system or base editing system can utilize that kind of purposes. Several mutations at the same time for the treatments. Sorry. Sorry. Maybe not.
In the time of starting.
Yeah, but if not, it's okay.
Well, no,
It's okay? Can we? Thank you.
just go forward.
Yeah. Sorry for this. Sorry. Yeah, please go ahead.
What do you want to be?
To the final, yes, I will do. I will do. I will do. So I just summarize like this, depending on the systems, most suitable editing systems are different. So mammalian and the plant and the yeast E. coli, that most suitable system shown by the red circles are different. So like this, by designing the system, you can create a very efficient mutation systems. Sorry. So I just would like to add one thing. This is, I'm not quite sure you are interested in, but anyway, by choosing the glycosylase, DNA glycosylases, you can create kind of random mutation systems. A glycosylase is probably, you understand that it's removed the base. So in a sense, after removal of the base, a variety of the repair reaction can happen. So like this, instead of the deamination, you can utilize another types of the DNA modifying enzymes. So actually, like this, if you utilize the glycosylase system, this window is 200 base pair. So that means that around 400 base pair, you can create a C2DC. This is a cytidine DNA glycosylase K. So you can create this kind of in a very wide range, and you can create random mutations. So if you analyze the mutation spectrum, very sorry, it's very small, but the C or Z to ATG change, we can introduce this kind of change by utilizing the glycosylase systems. So like this, not just the deaminase system, but also we can utilize another type of DNA modifying enzyme systems to create different type of mutations. So if I summarize the systems, a base editor creates point mutations in a very targeted way. And the base editor is very mild and controllable and very clean mutations. And the base editor works in a variety of organisms. So, and multiplex editing is possible because it doesn't cut the DNA, so it's very mild. And the endogenous deaminases, we can utilize that for the editing. So like this, this system, probably I hope I convince you that this systems is very, have many merits. So we have, created a company like BioPalette, which has a whole right of this kind of the base editing systems, and try to apply this one for bioproduction, agriculture area, drug discovery, and therapeutics. So hopefully this company also collaborate with some of you. So thank you very much for your kind attention. Thank you very much.
Thank you. Thank you very much. We have time for 2 questions.
Hi, great talk.
Yes.
I was wondering, what are your takes on the recent paper published, genome-wide or transcriptome-wide cytidine demethylation at the RNA level?
Yeah. I think it's possible. I think so. And also genome scale scanning of the genome itself also. We can do the genome scale scanning. We are now doing that.
Yes.
My question relates to safety.
Yes.
I think considering that you show that expression of AID,
yes,
regardless of Cas9, can also induce base editing, only
yes,
dependent on unwinding of the DNA. I'm thinking about transcription bubbles or any non-specific. Yes. What do you see over there?
Yes, so far we of course did whole genome sequences after editing, so we cannot find out some specific off-target, so far. So in a sense, it's actually not so high, I think, off-target levels, and that is because probably because the AID is just active for the single-stranded area. But of course, what you mean, there are many single-strand, could be tentative, because of transcriptions, there could be single-stranded. But we think because, the AID is, just attached to the DIET or Nikkei's Cas9. So AID is fixed to the specific bubble. So to each created by the guide RNA. So that's the reason why I think this is very specific editing systems.
Thank you.
Okay, thank you very much for the speaker.